Today, I'm going to start a course to teach experimental biologists how to do basic machine learning. So why might an experimental biologist need to know anything about machine learning? Well, I think this will help you formulate your research questions, design experiments that are informative, help you make the most of the data that you already have available from previous experiments or from existing publicly available data sets, and it'll also help you effectively communicate with bioinformaticians and other collaborators. Most importantly, machine learning can be done without access to a lab. In this mini course, I want to first help you learn the core ideas and principles, and only afterwards teach you how to apply them to your work. I want you to be able to mentally visualize how the algorithms work, even if you don't really have much of a background in either math or programming. So, what is machine learning? Well, in machine learning, you use data to train a model that can make predictions of an output given some input variables. And machine learning is the process of finding a good model using training data. Before you begin, you'll need to have an idea of what kind of model you want to build. So you need to decide what input variables you'd expect, what the predicted output would be. Also, you should consider what data you have to train the model. And finally, maybe most importantly, you need to think of a good criteria for evaluating whether your model is good. Let me give you an example of how biology can apply machine learning techniques. So, for example, let's say that you have a research project where the aim is to diagnose cancer from images of cells. So, before you begin, you need to think of your input variable, which in this case would be an image of a cell. In essence, you want to be able to give your model an image of a cell and have it make a prediction of whether those cells are cancerous or normal. So your output variable is a categorical label, in this case, cancer or normal. To train your model, you might have a collection of thousands of images of cells that have already been labeled as cancer or normal by experts. Finally, you might want to build a model that is as accurate as possible at predicting whether an image is of a cancerous cell or a normal cell. This framework is a little different from what you might expect given the kinds of statistics you learn about when you're studying biology. For example, in the classical biostatistics framework, you might ask a question such as, what characteristics are significantly different between cancer cells and normal cells? And then you'd go through different characteristics, and for each of those characteristics, you'd come up with a p-value indicating whether or not those variables are significantly different between the two categories. For instance, you might ask, is cell size significantly different between cancer cells and normal cells? Is cell shape significantly different between cancer cells and normal cells? And so on. In the end, you end up with a list of different characteristics and p-values associated with each. And from that, you can identify a list of characteristics that are significantly different between the two populations. However, you do not know whether these characteristics are sufficient for accurately diagnosing cancer. In this example, cell size is not sufficient for accurately diagnosing cancer, even though cell size is significantly different between cancer cells and normal cells. You can see that the distributions of cell size are very different from cancer cells and normal cells. However, there's a wide range of cell sizes where there's overlap between the two distributions. And if you found a cell that was in this range, you wouldn't be able to make an accurate diagnosis. In machine learning, we phrase the scientific questions in a slightly different way. We might ask, how can we best predict whether an image is of a cancerous cell? Then, once we build a model to make predictions of whether the cells are cancerous or not cancerous, we can look at the model and try to identify which characteristics of the image helped us make the best predictions. In the end, we do not find all the characteristics that are significantly different between the two groups. In this example, nucleus size and cell size are both variables that are significantly different between the cancer cells and the normal cells. So you can imagine coming up with a model that exclusively uses nucleus size to distinguish between cancer cells and normal cells. And this model, basically you'd input at the nucleus size, and if it's above this threshold, you label it as a normal cell, and if it's below this threshold, you'll label it as a cancer cell. And this model would be 100% accurate. However, an alternative model, which would be just as accurate, would be completely based on cell size. In this case, if the cell size is higher than this threshold, you'd label it as a normal cell, and if it's lower than this threshold, you'd label it as a cancer cell. Therefore, nucleus size and cell size contain redundant information. It's equally likely that the machine learning model will select one variable but not the other for making the prediction. So when we look more closely at the model to see which variables are being used for making the prediction, we might see that nucleus size is used for making the prediction, but not cell size. However, this does not mean that cell size isn't significantly different between the two groups. Let's look at another example. In this case, we want to predict which wheat lines would produce the greatest yield. So now we need to think about what is the input and output to the model that we want to build. Let's say that the inputs are the genetic variants, i.e. the SNPs, in a line of wheat. We could input that into the model, and that could produce a prediction of the final yield. To train the model, we might have thousands of lines of wheat, and for each of those lines of wheat, we might have its genotype and the yield measurement. Then we might want to select the model that minimizes the mean squared error. And since I promised you that you wouldn't need to know any maths for this course, let's just say that this is kind of like an accuracy for continuous data. Again, we can look at the difference between how the scientific questions are phrased in classical statistics versus machine learning. So in the classical statistics that you might be aware of, you might ask which genetic variants are most significantly associated with high yields. And then you'd go through each of the genetic variants and see if they're significantly associated with a yield. In the end, you get a list of SNPs that are significantly associated with yield, but again, you do not know how well those SNPs can predict yield. In machine learning, the question we'll ask is how can we best predict yield from genotype? Then, once we build a model that is as accurate as possible at predicting yield, we can look at which SNPs helped us make the best predictions. Again, we do not know if we'll find all of the SNPs that are significantly associated with high yield. Also, just like in the other case, we do not know if these SNPs are causal. All we know is that these SNPs can be used to predict yield. Let's look at a third example of how to use machine learning in experimental biology. In this ecological example, we might want to identify distinct populations of bacteria found in a soil sample. And the aim is to predict which bacteria belong to which population. The inputs might be the genetic variants in the bacteria, and the output of the model would be a population label. However, we might not know ahead of time what these population labels are. To train the model, we might have lots of examples of bacteria SNPs from the soil sample. However, we might not have population labels because, again, we have no idea what the population labels are. In this case, well, we can't pick a model that is as accurate as possible because we don't know what the right solution is. However, we might want to define population labels as good if each member of the population is similar to one another, but very different from members of other populations. So what will we cover in this course? We will cover supervised learning and unsupervised learning. Supervised learning is when the output variable is known, like in the cancer diagnosis case and in the wheat yield case. In unsupervised learning, the output is unknown, like in the bacterial ecology case. We will also look at categorical outputs and continuous outputs. Categorical outputs are discrete labels like cancer and normal, or the population group. Continuous outputs are things like yield or height or weight, things that are numerical. This is how the examples we've looked at map onto these squares. Each of the different squares have a unique name, and these are the techniques we'll cover in the course. We'll cover classification techniques, regression techniques, clustering techniques, and dimension reduction techniques. So today I've introduced machine learning. The next series of courses will be about supervised learning, then unsupervised learning, and finally, I'm going to have a course about forming a coherent biological narrative by combining different supervised learning and unsupervised learning techniques. Before we end today, I would like to briefly discuss how I'm imagining the data to be structured for the remainder of the course. I imagine that you've put your data into a table that looks something like this. Each row represents an observation. Each column represents a variable that you've measured from an experiment. In unsupervised learning, all of the columns are going to be the input variables into your model. For supervised learning, one of the columns, it doesn't have to be the last one, but I'm just showing it here for ease, is the output variable. Mostly, when I'm describing the different algorithms, I'll assume that the input variables are all numerical, and that the output variable is numerical in the case of regression, or categorical in the case of classification. You might be concerned because it could be that you have data that doesn't seem to easily conform to that kind of table structure. However, you don't need to worry because usually there's some way of transforming your data into a row of numerical input variables. For example, if you have an image, every pixel actually has 3 numbers associated with it, indicating how red, how green, and how blue that pixel is. You can take those numbers and flatten all those numbers into a single row of values. Also, you might have sequence data like DNA sequence, RNA sequence, or protein sequence. And this can also be turned easily into a row of numbers. You can start off by filling out the following table, where each row is a position and each column is a letter in your alphabet, like in this case, A, C, or G. You put a 1 if the letter in that position is the same as the column heading. So in this case, A is the first letter, so you put a 1 here and you put a 0 elsewhere. Note that even though there's 4 letters in the DNA alphabet, we only need to have 3 columns, because if the letter is a T, you can just put 0 in each position. This means that for every base, you'd still have a unique combination of 1s and 0s. You can flatten this into a row. So for example, starting from here, you go 1, 0, 0, 0, 1, 0, 1, 0, 0, 0, 1, 0, and so on. What about missing data? So for example, let's say you're trying to build a model to predict the future health of a group of patients based on blood pressure, BMI, amount of exercise, age, and a bunch of other variables. But let's say that in your training data, you don't have the blood pressure measurements for a small number of patients. Many machine learning strategies do not, by default, have a clever way of dealing with this missing data. However, there's various strategies for substituting the missing value with a reasonable number that will hopefully have as small an impact as possible on the final model. This procedure is called imputation. So before the next course, I'd like you to think of some data that you have collected as part of an experiment. Is the data arranged in a suitable table like the one I've described? If not, can you think of a strategy for rearranging the data to be in a suitable table format? Think about how many variables you have and how many observations you have. Also, think if a particular column that you have would be suitable as an output variable, as this will help you decide whether supervised learning or unsupervised learning would be the best approach for your research question. Then, I'd like you to think more deeply about the research question. Try to state the question from a classical statistics perspective, and then rephrase the question from a machine learning perspective. What are the differences between what you'd learn using each perspective?